And welcome to Dimensionality Reduction tutorial number 1. In this first tutorial, we'll cover the basics of dimensionality reduction, and then the second on Thursday, we'll cover some more advanced techniques. But before I go further, of course, I want to thank the BCS Seminar Committee for organizing this tutorial series, and Chris for performing the audio-video that will go with it. So if you miss anything, don't worry. Or if your friends aren't here, that's OK, because they can go online and see all the materials along with the PSET after the seminar. So in this presentation, we're going to go over some definitions and examples to begin with. Then Emily will take us through a deep dive into the math and the Matlab behind one of the most important dimensionality techniques we have out there, principal component analysis implemented as SVD, or singular value decomposition. Then we'll conclude with some reflections on dimensionality reduction, what it can and can't do for you, as well as the broader space of other techniques that are out there. And finally, we'll have our traditional PSET office hours time at the end, which will be the last hour or so, where you can work on the PSET or ask questions or just discuss with other people what techniques work for them for dimensionality reduction. Now, dimensionality reduction begins with data, as all good things do. And your data can take many different forms. It might be images, for instance, where you have all these pixels. It might be brains, since we're all neuroscientists. And it might be these voxels. Or it might be these giant feature matrices that you're using for machine learning, and you just want to try and take 100,000 different features and put them into a classifier. So what can you do with all these things? Well, every number in that image or every number in that brain full of voxels you can think of as a dimension. So oftentimes the first step in doing dimensionality reduction is to unspool all those numbers into a nice long vector like you see here. So if you had an image that was 4 pixels and you had multiple images, you might take that first image and use Matlab to reshape it as a vector. But don't be fooled. Your image or your data doesn't need to begin out as one of these 3 things. It might also naturally come as a vector. For instance, these might be a time series of microelectrode recordings. And each one of these time series might be a different task. For example, a direction and a reaching task, where the first time series, monkey reached to the left. Second time series, monkey reached to the right. So that's great. But you know, if you want to visualize this data, for instance, it's very hard to do if you have 4 dimensions to deal with. Matlab, unfortunately, does not offer 4D plots so nicely as it does 2D. So you want to go from 4 dimensions to 2 dimensions. And you can do that in a number of different ways, one of which we'll cover today, and one of which you see here, which is you can throw out the other 2 dimensions. Obviously, there are reasons to do that on some cases, but most of the time you want to go from here to here in a principled way. So dimensionality reduction is the idea of throwing away some data in cases in order to make the knowledge or the structure of your data come out and be more visible. Now, visualization is, of course, one of the most obvious reasons to do dimensionality reduction. You can't visualize a 100,000 dimensional matrix as well as you can 2. So you might do dimensionality reduction for visualization purposes. But you can also do it to discover structure in your data. For instance, in this lower left-hand picture, you see all these different A's. But you don't really need all those pixels for all those A's to understand what's going on. You just need one A. And if you're sharp, if the projector is good enough, you can tell that the difference between any of those A's is the fact that they've been scaled and they've been rotated. So it's kind of like there's this 2-dimensional transformation that's going on here. Another reason to use dimensionality reduction is for data compression. For instance, bandwidth is a limited resource, and you need to make sure you get as much important information as you can over that channel. And finally, you might use dimensionality reduction for noise or artifact detection. Not everything that you've collected is actually useful for you. Wouldn't it be great if there was a way to automatically separate out the good stuff from the bad stuff? And in this background are a whole montage of different techniques that have been applied, including songbird data, the MNIST data set, which is a set of all the digits. And you can see after dimensionality reduction, all those pixel images actually end up grouping into the numbers that you would naturally expect, which is a really great result. At this point, I'd like to hand it over to Emily, who's going to talk and take us through a particular dimensionality technique called principal component analysis.
Cool, thank you. OK, so as Greg introduced, when you have data, you have some choices about how to represent it. So let's say that this is some data that I recorded, and I took 2 recordings. It could be positions of an animal in a 2D arena. It could be recordings from 2 different electrodes, from 2 different voxels. But each point, I made 2 measurements, and I'm plotting it in 2D space. And so now I can represent that data in a matrix where I have n dimensions, in this case 2 that I've recorded, and I also have p data points, in this case 1,000. So that's how, in a matrix, is how we usually represent data. But this choice of 2 measurements that we made might be somewhat arbitrary, and we might be interested in something else. So for example, we might be interested only in how far our each data point lies along this line. And so we can call this one basis for the data, the original basis I recorded it in, and then this is a new basis, which you can think of as just taking this original basis, rotating it, and then forgetting one of them. And so now I can represent each data point either as these 2 numbers in the original basis, or as one number, which is just its position along this blue line. So that gives my whole data set 2 matrices, either this larger matrix in 2 dimensions, so 2 dimensions per point and p data points, or this matrix where every data point has just one number associated with it. And how do I go between these 2 different representations? Well, one way that I can do it, if I have just the information from of where along this blue line my data point sits, then I can estimate where it was in the original space by looking at its projection onto this line. And so in matrix notation, you can look at the blue vector in your original basis and take each data point, project it onto that new basis, and that will give you an estimate of where that data point is in your original basis. So this is a framework that is common to a lot of machine learning techniques, including PCA, but also many techniques where these might be different features about your data, different clusters, different basis elements or dictionary elements. And what you're doing in these machine learning and dimensionality reduction techniques is often taking a large matrix of data and rewriting it as a multiplication of 2 new matrices, one of which is your recoded data points. So in this case, the position along the blue line. And then another is this new basis, this translation between your recoded data and your original data. So what PCA does specifically is it finds the direction of greatest variance in your data and uses that as your new basis. And it does this by calculating the eigenvectors of the covariance matrix. So if you saw this data, you would see that this is the direction of greatest variance in the data, and then PCA extracts that and rewrites your data in a new way as the projection onto the first few directions of greatest variance. Okay, so I'm going to go through an example of data from monkey motor cortex in a task where the monkey was reaching in 8 different directions. So on each trial, he's given an instruction queue, and he'll reach in one of 8 different trials. So this data can be represented in a matrix. For now, I'll be talking about an example where you're recording just from one electrode, but later in the exercises, there's a cool example of more than 100 electrodes, and you can pull out this reaching structure in that as well. So here, in this big data matrix, I have each point is n dimensions. So there are n time points for every trial. I'm measuring the, or this data was from Georgopoulos' lab, but I'm measuring the time, the activity of every neuron throughout an entire trial. So I'm saying, OK, starting at the go cue, what was the spike rate at every time in through the whole trial? And there are 158 different trials in different directions. OK. So this is what the data looks like. It's a matrix of time steps and trials and colored by the rates. So these rates are organized according to the different directions. And so these will all be similar directions around in a circle. And you can sort of see some structure here. So you can sort of see that the neuron fires less for some of these trials, and then more for these, and then less again for these. Are there any questions about the data? No? We're good. OK. Cool. So what are we going to do with this data? I'm going to show you a Matlab example of doing PCA on this data to extract some structure in it. OK. Cool. So first of all, I just load the data into Matlab, and I plot the data using image scale. So this is the plot that I showed you in the slide. I've just plotted the data. Next, what I do is I subtract the mean from the data. So I say for every time point, subtract the mean activity of that neuron across all trials. And this is useful so that you get your cluster of data points all centered around 0. OK, so here I'm subtracting the mean of the data. And this matrix is the same matrix but mean subtracted. So now what I'm going to do is I'm going to compute the covariance matrix of this data. So I'm going to look at, for different time steps, which time steps are vary together. And I will do this by, so Z is what I've called the mean subtracted data. And Z times Z transpose is proportional to the covariance matrix. So let's see what that looks like. So this is the covariance matrix of time steps by time steps. And you can see here that, first of all, neighboring time steps tend to be doing similar things. So this is like across all trials, neighboring time steps are highly correlated in yellow. And then you can see time steps, let's say around 10, are anti-correlated with time steps around this time. So it'll start to move, and then a little later, the neuron will be doing something different. So in order to do PCA, we take the eigenvectors of this covariance matrix. And when you're taking the eigenvectors, you want to know how much variance was in each direction. So the eigenvalue of the first eigenvector of the covariance matrix will tell you how much variance in your data was along that direction or the next direction or the next direction. So that's what I'm plotting here. I'm taking eig of the covariance matrix, and then I'm plotting the eigenvalues. And you can see that the first few eigenvalues are higher compared to the rest of them. And so that's motivation for saying much of the variance of your data is explained just by these first two eigenvectors. So what does the data look like projected into these first two eigenvectors? Let's first look at these eigenvectors themselves, because these eigenvectors are vectors in the time step space. So here I can plot time steps, and then I can plot the coefficients of these first two eigenvectors, these first two principal components. And I can see that one of them is high in the early times and then lower for the rest of the trial. And then the other one starts low and then gets high, low and high again. And so how to interpret this would be that any trajectory that you see the neuron go through would be mostly explained by a combination of these two things. So for example, on some trials, you might see the neuron mostly doing the blue thing. And on some trials, you might see the neuron mostly doing the red trajectory through time. And other trials might be a combination of those two things. But it can mostly be rewritten as a combination of just these two patterns. So let's do exactly that. Let's rewrite our data just as a combination of those two patterns and forget all the other variants in our data. So what I'm doing here is I'm rotating the data into the principal component space, and then I'm dropping off everything but the first two principal components and then rotating it back into the original space. So everything that wasn't one of these two first two principal components, I'm just forgetting about. So this is just the original data again. And then this is what it looks like filtered to just those two principal components. And you can see that it looks sort of like a denoised version of the original data. And you need to think about with your data, when are you, by doing this, extracting the signal, and when are you extracting the noise? But this is what, in this case, it looks like when you filter by just keeping the projection of your data on your first two principal components. OK. So are there any questions at this point? No? So we made a choice when we did this. We made a choice to calculate the eigenvectors as time courses throughout the trial. But we could have chosen instead to look at a flipped version of our data matrix and calculate our eigenvalues in the trial dimension. So this is just a flipped version of our original matrix, where now the time step throughout the trial is here, and the trial number is here. So I can compute the covariance matrix of that matrix, and now this gives me the covariance between different trials. So it says that trials that had similar directions are highly correlated with each other. This might not be super interpretable just looking at it like this, but this is, you could see it's higher here, which means that these trials might be similar to these trials up here. It's similar patterns of activity for those two trials. OK. So we can do exactly the same thing as we did before and compute the eigenvectors of this matrix, doing it this way, and see what it looks like. So the first thing we notice when we compute the eigenvectors and eigenvalues is that the eigenvalues look exactly the same as they did when we did it the other way. So every eigenvector that we computed in the time step domain has a corresponding eigenvector in the trial domain. And so you can think about that as that the time step trajectory could happen on certain trials, and it'll happen on some combination of trials. And I'll get back to that in a little more detail. OK, so what do the first two trial eigenvectors look like when we plot them? This is what they look like. And might be a little noisy, but you can see that for some of the first trials, the red and the blue are sort of doing different things, and then they might flip at different moments. And so these are basically what trials are correlated with each other. And you can say that different trials would have similar loadings on each of these principal components. OK. So what does it look like when we filter the data just using the first trial principal components? This is the original and then the flipped original matrix. And then this is what you saw before, filtered by the time step principal components. And now this is the flipped one, filtered by the trial principal components. And you can see that these are just flipped versions of each other. So that's useful. It's done a very, very similar thing by, it's filtered it basically in the identical way. And so why is it so similar when we do it in these two different ways? It seems like we've done something on exactly the flipped matrix, but we're getting the same filtering, we're getting the same eigenvalues and everything. It turns out that this isn't a coincidence, and that PCA, when you do it this way, you can actually rewrite your original matrix as this singular value decomposition. And the singular value decomposition rewrites your original matrix. Let me go back to the slides. Sorry. It rewrites your original matrix as a combination of 3 new matrices. The first one will be your time step basis. The next one will be just your eigenvectors. And the third one will be your trial basis. And so another way of rewriting this is that your original matrix is just a sum of new matrices, which are the first U and the first V. So the U is the first time step principal component, and the V is the first trial principal component. So this matrix is what that first time step principal component was doing over all the trials. And I can show you a picture of what that looks like. And you can see that that reconstructs your original matrix. So let's see. I'll go to this here. So here's the original matrix. And then here is the first time step principal component outer product with the first trial principal component. And so this is the second time step principal component with a second trial principal component, third and fourth. And your original matrix is just a linear combination of different weights of these matrices. Okay. Do we have questions there? No? So anyway, that is basically done with that, but I think it's pretty cool that you always have these choices that you're making of which way to decompose your matrix. But when you make this choice with PCA, you wind up getting the same eigenvalues either way you do it, and the same decomposition either way you filter it. Cool. And in the exercises, you can look at similar data with monkey reaching, and you can actually see if you plot in the first 2 principal components, you'll be able to see the 8 different directions clustered together in a circle. So they're cool exercises. You should try them.
So thanks. So we've seen this really powerful technique, principal component analysis, implemented as singular value decomposition. This is one of the most fundamental ways of performing dimensionality reduction that we've gone through. You can see that there's a number of ways of attacking it in Matlab, very friendly interface for implementing it, only a couple lines of code. But you know what? When I first did a PCA, I thought this is the best technique ever. And then I found out it can fail. I thought, how can PCA fail? Well, implicitly, the idea behind principal component analysis and these other dimensionality reduction techniques is that we're going after something that's fundamental about the data. We're trying to emphasize something. Well, we like to think that PCA is emphasizing the internal structure of your data, making this really obvious. But as you are following along with Emily, we've seen that really PCA is getting at something else. It's getting at the variance of the data. Now, if you give your PCA algorithm data that looks like this nice cloud that you've got here, PCA will say, OK, well, this is good. I got a cloud of data. And you know what? There's a lot of data points along this direction. So that's going to be my first eigenvector. And what's the direction where there's the second most amount of variance? Well, that's going to be along this direction. So they're orthogonal. That'll be my new basis. Here you go, Greg. And Greg will say, well, I want to do dimensionality reduction, so I will take the first eigenvector because that has the largest eigenvalue, the most variance in my data. That's great. So I'll throw out the information along the second one, and I'll just keep the position of the point along this long arrow. And that's all well and good, except that I knew something else about my data that PCA did not know. In fact, my data came from, say, two different classes. Monkey reaching to the left, monkey reaching to the right. For instance, left would be the red dots and black would be the right dots. And if I throw out the position of those dots along the second eigenvector, because I'm doing dimensionality reduction down to one dimension, what I've done is I've collapsed all these dots that are red and black on top of each other. But that's not at all what I wanted to do. I wanted to separate these directions. To separate them, I should have, in fact, been looking at the position of the dots along the second eigenvector, not the first one. So PCA didn't know this, and if I didn't know any better, I would just say, well, my data is garbage. You know, I've done dimensionality reduction, nothing here, to go back to the lab. But this just gives you a sense of the fact that PCA is an unsupervised technique, meaning it just looks at the data as you give it. It's not aware of this labels that go with your data. Fortunately, people are aware of this problem, and there are other techniques. PCA is just scratching the surface. Dimensionality reduction is a huge field. If you go on Wikipedia, there's something like 30+ techniques and many, many papers. And there's different ways you can look at all these different techniques. One of the most obvious is what we just talked about, the division between supervised techniques and unsupervised techniques. Unsupervised techniques like PCA or SVD are not aware of this extra information that goes with your data. Whereas supervised ones specifically are saying, he wants to separate left versus right. What can I do that will reduce the dimensionality of my data and at the same time preserve the separability that I'm going for? There are other ways of looking at these techniques too. There's linear versus nonlinear techniques. Linear techniques, very generally, are this idea that Emily touched upon where we took our data matrix and we broke it into all these other matrices. Well, if you keep multiplying matrices, that's linear algebra, so it's kind of like a linear technique in a series of linear transformations. But there's a whole other family of techniques, of nonlinear techniques, in fact. What they do is they say, well, I've got these two data points. Each has 100,000 dimensions associated with them. And I think they're close in 100,000 dimension space. But I can't visualize that so well. So what I want to do is put them in two-dimensional space, but still keep together points that were close in 100,000 dimensional space and two-dimensional space. And what you can do is run various kinds of optimization algorithms that say, well, these 2 points were close up here. If I do this kind of transformation, they'll actually still remain close, and that'll be great. And so there's a whole slew of techniques that will do that. You can think of those as nonlinear techniques. And then the final point here is kind of subtle. I wasn't aware of it myself for quite a while. And it's this idea between techniques that will do dimensionality reduction and let you apply that same dimensionality reduction transformation that you came up with to new unseen data versus other techniques which are in fact just really good at visualizing the data. So for instance, if you have your data matrix and your monkey data, and you do PCA or SVD, and you get these eigenvectors, that's great. And you know you want to keep only the first, whatever, 2 eigenvectors. And then someone gives you some new data that you kept back from this monkey. And you want to see that new data in your two-dimensional space. Well, you actually don't need to stick that data onto your giant matrix that you already had, run PCA again, get out the new eigenvectors, and project the data once more. In fact, you can just reuse those eigenvectors that you already computed and just do a simple dot multiply in Matlab to get back the lower dimensional data. However, if you run some of these very powerful nonlinear techniques, by adding a new data point to that giant matrix, the algorithm actually has to rerun sometimes in order to figure out, well, where does this go? I don't know, because it's all in relation to all these other points that you have floating around. So it's just something to be aware of, and it's something that certainly people have tackled. There are what's called out-of-sample extensions for some of these other techniques, and you can look at algorithms for that. But again, in this tutorial, we're showing you PCA and SVD is the simplest unsupervised linear technique that lets you do mapping and visualization. So you can always do visualization. So in summary, we've talked about dimensionality reduction, this idea of having lots of data in very high dimensions, and wanting to toss away some of that data in an intelligent way in order to emphasize the information or structure of your data, whether it be for visualization, data compression, or artifact removal. We've covered PCA and SVD, these two very powerful techniques, kind of two faces of the same coin. The idea that you can take your big blob of data and split it up into matrices, and each of these matrices might be more or less important depending on the eigenvalues. And of course, PCA and SVD are just two of a huge variety of techniques that are out there that go by many different names. So with that, I just want to give a small plug for what's going to come on Thursday, which is one of these more advanced techniques, independent component analysis. You might see this used a lot for MRI data. And finally, we have here some readings on some software. So these are some excellent PDFs that go through not just PCA and SVD, but also a number of other techniques, both linear and nonlinear, the pros and cons. And of course, software. So if we have MATLABers in the audience, of course, there are MATLAB toolboxes out there for dimensionality reduction, but there are also Python toolboxes as well. I encourage you to check out scikit-learn, for instance, if you're interested in more information on that. With this, I'd like to close the presentation part of our discussion on dimensionality reduction. I'm happy to take any questions. Otherwise, Emily and I will stick around for one-on-one questions or small group questions while you work on the PISA if you'd like. So thank you all for your time. I hope this might be useful in your work.